Okay, here's another AQA required practical, and this is food tests. Okay, so there are tests for each of the three main food groups, which of course are carbohydrates, and that includes sugars and polymers such as starch. You know, so they're quite different things to look at and to taste, but they are the same things. They're both carbohydrates and proteins. And finally, fats or lipids, depending what you want to call them. Okay, so let's do the first test, which is iodine. And this is a test for starch, which is, of course, a complex carbohydrate. It's a polymer of glucose. All right, now, this is an easy test. You get a sample of the food, usually best to put it onto a spotting tile. And then you get iodine solution, which is this brown color here. And you put a couple of drops of iodine solution onto your food. If you have starch present, it goes black. Okay, it's actually bluey black, very dark black color. So the kind of foods that will give you a positive test for starch, if you a lot of cereal grains, so such as rice, anything made from rice or cooked rice, anything made from wheat, so that's going to include bread and flour. And pasta, and things like potatoes and carrots. They contain a lot of rice, and apples as well. Quite a lot of starch in apples. If you test apples and potatoes, and it doesn't give you a really good clear black color quite often if it's raw, because the iodine can't get inside the cells where the starch is. So it might be best to just give it a bit of a crush with a mortar and pestle, those ones. But if you're testing, you know, rice or wheat or whatever, then it will go black almost immediately. Okay, so it's a very easy test and very clear-cut result. Right. The next one then, this is Benedict's test, which is a test for reducing sugars, and that usually means glucose. Glucose is a reducing sugar, but it's not sucrose. Now, sucrose is the sugar that you put in your tea and you use making cakes and things like that, and is added to food to make it taste sweet. But sucrose is not a reducing sugar, so it won't test positive with Benedict's. Lactose, which is present in milk, is a reducing sugar. That will give a positive test with Benedict. So Benedict won't show the presence of all sugars, just reducing sugars. And if you know what reducing means, well, in chemistry, you know, it means the sugar is actually acting as a reducing agent, but don't worry about that. Okay, so how do you do Benedict's test? Here's Benedict's test, and it looks that turquoisey color because it contains copper sulfate and a bit of sodium hydrogen carbonate. And then you put in your food to test there. So you can either add a liquid, or you're testing like a bit of Coke or like fruit juice or something, or you can put a bit of solid food, but you should try and squash it up in a mortar and pestle first, because if you put in something which is like a bit of potato, the Benedicts can't get inside the cell, so you need to squash it up to kind of release the contents of the cell. Right, so then you heat it up in a water bath for about 5 minutes or so, and then you'll notice the first sign, if you have got a reducing sugar, the first sign of a positive test, it goes a bit cloudy. So that means you've got a precipitate forming. It's insoluble. And it looks, first of all, it looks green, and then it becomes more of an orangey color and finally becomes a red color. But they're all precipitates. They're all solid. So you're looking for cloudiness. And if you've got very low levels of reducing sugar, you probably just see this green color. Whereas if you've got lots of it, then you'll definitely see the red color. But so it's kind of semi-quantitative, but you can tell from the color it goes. But any cloudiness, any precipitate form is indicative of you've got a reducing sugar present. If it just stays perfectly clear, turquoise color, no reducing sugar there. You can test any kind of food you like on that, so you can put little bits of meat or rice or anything like, but they won't contain sugars. So it's mostly things that will taste sweet. So like fruit juice, again, no fruit juice may contain sucrose, not glucose. So even though it does taste sweet, it mightn't give a positive test. But you can also test a bit of milk, because milk contains lactose, and that should give a positive test. Foods such as meat and so forth, they don't contain sugar, so they'll probably give you a negative test. Right, next one is Biuret Test. This is a test for protein, okay? And this is, to make this work, the protein has to be kind of solubilized a little bit. So you can't just chuck a piece of raw meat in and expect it to work. You would need to bash it up in water with a bit of with a mortar and pestle, with a little bit of water, and make a sort of semi kind of suspension of it before you try it. Biuret, sometimes you get it given ready to use, ready to go, and it's this dark blue color, but quite often people, that goes off, so people make it up just before they're going to use it, and that makes it, you mix Biuret solution A, which is actually copper sulfate solution, that turquoise color, and Biuret B, which is just an alkaline solution, and it goes this darker blue color here, and that's ready to use. Then you add your food sample and say it should be a little bit solubilized, a little bit bashed up and made into a sort of suspension, and leave it. You don't need to heat it, you just leave it, and if you get this nice nice mauve color or lilac color, that shows you've got some protein present. So you can test egg yolk contains protein, egg white contains protein. If you test things with a lot of starch in, like rice and pasta and potato and stuff, they usually give you a negative test. They don't contain a great deal of protein. They do contain some, but not enough to give you a positive test. If you get a little bit of cooked meat, cooked is better, so it will release the cell content and ground up with a mortar and pestle. That should give you a positive test as well. Certain grains, certain seeds like peanuts, there's quite a lot of protein in them. That should give you a positive test as well. Finally, we have got the test for the ethanol emulsion test, which is a test for fats or lipids, depending on what you want to call them. Okay, so what you do there is you put the food in the bottom of the test tube, and first of all you add a little bit of ethanol. Now lipids, fats, as you know, they're not soluble in water, so you try and mix olive oil and water, they won't mix, they just separate into two layers, but they are soluble in ethanol, which is alcohol. Okay, so and by the way, you should, with ethanol, you should do it after you've done your Benedict's test, because you don't want any naked flames, because it's very flammable. Add the ethanol here, and that will dissolve some of the lipid. It probably won't look like it's dissolved, but it has dissolved a little bit of it. Okay, and then what you do is you add water, and the dissolved lipid will come out of solution because it won't dissolve in water, and it will come out in solution as here you'll see tiny droplets called an emulsion. It's tiny droplets of fat, and that tends to look cloudy. So the positive test, if you get any kind of cloudiness, then you've got a lot of lipid in your sample there. So if you test things like potatoes and starchy foods and pasta and rice and things, they don't contain any lipid. You won't get any there. Meat, even lean meat, contains quite a lot of fat, so that should give you a positive test. If you test egg white, there's not much fat in that. That won't give you a positive test, but egg yolk, there's a lot of lipid in that, and that will work. Peanuts and some seeds like sunflower seeds and so forth, they also give you a positive test. Right. Just finally, an alternative test for fats, which works really well and is much easier, is it's called the grease spot test. And all you do is you just rub, like if you get a peanut, for example, with a lot of lipid in it, you rub it on a piece of paper, you'll notice it leaves a translucent sort of clear mark on the paper. You probably notice this when you've been eating crisps or something, and then you touch your paper. You've got lipids, you've got fat on your hands, and it makes greasy marks on the paper. So that's the grease spot test. It's a pretty easy way of testing for fats.